Hello everyone, my name is Megan Sanford, and today I'll be going over techniques to analyze diets in sea otters. The problems associated with the analysis of sea otter diets stem from their aquatic lifestyle. This lifestyle makes it difficult to apply traditional terrestrial diet study techniques. The techniques that are currently used also lack practicality. This is in terms of understanding the full comprehensive diets of sea otter populations. The techniques in this review are direct observations, necropsies, sprint analysis, DNA barcoding, and stable isotope analysis. The first technique of this review is direct observation, which is done when researchers actively go into the field to observe what sea otters are eating. The pros of this method is that other information can be collected, including temporal and spatial data associated with what the researchers are observing, and therefore comparison between individual otters can be completed. This method is also passive and cheap, especially when compared to other methods within this review. The cons of this method is that it really is highly dependable on the viewpoint of the researcher and what they can actively see. Also, human presence may affect the behavior of the otters and therefore what they are selectively eating. And finally, many otter populations are difficult to get to places and therefore it may be impossible to trek through and therefore directly observe. The next technique is necropsies and postmortem analysis, which include collecting dead specimen and opening their stomach and viewing their contents. The pros of this method is that it's very common, and so standardization and comparison of research are relatively easy to do, and it's non-invasive as the animals are already found dead. The cons include that it's very limited sample size, there's varying digestion rates of the food sources that otters eat, the observational bias within different observers of what they may be seeing in the stomach contents, and there's lack of other information in this sample size, including how this otter may have died, which all therefore lead to sources of error and bias within your results. Sprint analysis, or SCAT analysis, is done by going into the field, collecting the sprint, and bringing it back into the lab for analysis. The pros of this method is that it can also be used as an index for population size, however there are many cons. First, data standardization is difficult. This method leads way into a lot of observational bias in which the individual observer is viewing within each sample. Also, frequency of occurrence, which is the most common method of this technique, has been proven to be not accurate. This was done by comparing what captive otters were eating and what their sprint analysis was showing, which were not the same. Therefore, this technique may not be a viable option for understanding the diets within otters. The next method in this review is DNA barcoding, which has historically been done in other otter species by looking at their sprint. The pros of this method is that it's able to quantify highly digestible food sources, and in otter diets, this includes mollusk. This is also a progressing technology, which means as time goes on, this will be only more efficient as a method for diet composition. The cons of this method include that sequence data needs expert knowledge, which means that new labs or current labs that don't have the funds to invest in this research may not be able to contribute to this research. Also, DNA is a highly degradable substance, and therefore this method is really dependent on how quickly you can get the samples back to the lab for analysis. The final method of this review is stable isotope analysis, which is done by comparing the ratios of stable isotopes of the sprint vertebrae or tissues of the otters to the known ratios of the stable isotopes of their food sources. The pros of this method include that the data allows for the comparison of individual otters. It also allows for the analysis of simulated diets, not just what is currently in the stomach contents. It also allows for maybe less field effort when compared to other methods, and it can also detect elusive foraging behaviors. The cons of this method include that the current technology doesn't allow for the ability to discern between different food sources within otters. This means that you may be able to tell if two otters have completely different diets, but not exactly what they are eating. And this method also requires previously known tissue of values for the stable isotopes. My recommendation for the analysis of the diets of sea otters would be to use a combination of DNA barcoding and stable isotope analysis techniques. These technologies are only increasing in efficiencies, and the cons associated with both methods are being addressed currently. Together, these methods would allow you to track individual sea otter diet composition while also being able to compare it within the same population or among different populations, which is critical for management decisions. Thank you for listening to my presentation, and I want to take this time to observe all the studies that were required to compile this review. Thank you again, and please leave any questions or comments below.